Hi, everyone. This is Jane Dixon Weber, and I'm the Director of Education and Support Services at the National Fragile X Foundation. And today we're going to have a webinar with Dr. Elizabeth Berry-Kravis on the NeuroNext clinical trial for Fragile X syndrome. I think that because we're recording the webinar, we're going to go ahead and get started so that we can respect everybody's time on this. So before I introduce our speaker, there's a couple housekeeping issues. Dr. Berry-Kravis is going to give a short presentation and then she'll take questions. Please use the question box on the side of your screen for your questions, and also use the question box if you have any problems hearing or any other aspects of the webinar. Now I'd like to introduce Dr. Berry-Kravis. She is the Professor of Pediatrics, Neurological Sciences, and Biochemistry at Rush University Medical Center in Chicago. She received a bachelor's in chemistry from the University of Notre Dame, her PhD in biochemistry, and her medical degree from the University of Chicago. She moved to Rush University Medical Center and established a comprehensive Fragile X clinic through which she now provides care and support to over 600 patients with Fragile X syndrome. She is internationally recognized for both research and clinical work on Fragile X syndrome and FAXTAS and the other premutation carrier conditions. Welcome, Dr. Berry-Kravis.
Okay, thank you, Jane. So today we're going to talk about the clinical trial that we're running, which is funded by the NIH through the NeuroNext network, which is why we keep referring to it as the NeuroNext Trial. And the NeuroNext network is a group of institutions across the country that are given infrastructure funding by NIH to run really novel trials in neurological diseases. And so this trial is a very unique trial. It's something that really is a new design that we're trying to do to look to see if a drug alters learning in people with Fragile X. And so what I want to do is kind of go through the background for the trial first, and then talk about what's involved in being in the trial. And I think the background here is really important because this trial is built upon a really large amount of basic science and information that we have on Fragile X. Okay, so our trial is called FX Learn. This is our logo, and the NeuroNext logo is on here too. So I don't need to tell anybody on this webinar about Fragile X syndrome. I mean, we know it's the most common known inherited cause of intellectual disability and autism. And essentially what happens in Fragile X is that the FMR1 gene is supposed to make an RNA, and then it's supposed to make a protein. And that protein, it turns out, is really important in neurons for them to function. In Fragile X syndrome, we get this big mutation of more than 200 repeats. So the gene gets methylated or shut down. It can't make the FMR1 RNA, and then we have no FMRP, and that leads to all the brain learning and thinking and brain connectivity problems that we see in Fragile X. So essentially, Fragile X results from loss of FMRP, and I think that's important to understand because this trial is really based on the mechanism of what FMRP does in the brain. So we all know we have a clinic consortium. We have multiple clinics that treat patients with Fragile X. We all know that we do a lot of supportive treatment, therapy, education, medical support, behavioral, psychopharmacology. But, we have some big surveys from Don Bailey's National Survey, for instance, that tell us that really it's only the percentage of people that are green here that think their medicines are really working well for their behaviors. So we have a lot of room to move in terms of treating Fragile X. We need better behavioral treatments. We don't have any treatments for cognition. This is a big burden on families. And so there's clearly an unmet need, and we really need to try to treat the actual disorder rather than just using supportive treatments for behavior. Not that those don't work, but that we want to do better. So this is a little deep into the basic science, but basically this trial is based on basic science. So the Fragile X mouse model and the mechanism of FMRP, really understanding what FMRP does in the brain is what creates a window to these new treatments. So we have a mouse that has no FMRP, just like Fragile X patients. When you look at the brain, it's a good model for human Fragile X. And through this model and other studies, we understand that FMRP is like an uber protein that regulates production of many other proteins in the brain connections and helps the brain wire and learn correctly. So FMRP regulates the changes in strength of a connection that you need when you activate a neuron or a brain cell. And it needs to be regulated precisely for these connections to mature and create learning. And so loss of FMRP in Fragile X leads to these immature connections and abnormal, what we call synaptic plasticity, which is the ability for a connection to change with learning and make it easier for certain brain cells to activate, so that once you've learned, you can do something faster and easier. And we know this from studies in the mouse where we can see that these long, spindly dendritic spines are characteristic of immature spines, and these are the ones the Fragile X knockout mouse has. And then the normal mouse has the much shorter, more mature kinds of spines. And this is a characteristic we know is also true of people with Fragile X. So there is this mechanism that I showed a lot of different conferences that was worked out over many years. And it shows that there's certain receptors, particularly some glutamate receptors called metabotropic glutamate receptors, and particularly mGluR1 and 5, which they activate protein synthesis, which is necessary for brain connections to remodel and to learn, and FMRP regulates that protein synthesis. So in this model, we have glutamate activates this receptor, which is present on most of the brain cells, and then we get protein synthesis being activated. But the way it activates protein synthesis is by kicking FMRP off the ribosome, which is the protein-making machinery, and then you get more proteins made, and those proteins regulate other receptors getting moved in and out of the cell, particularly the AMPA receptor, which is the workhorse of the cell that makes a neuron a connection stronger or weaker. FMRP is also made through this process, and it kind of acts as its own feedback inhibitor. And so when everything is working right, you have just the right amount of activation coming through here and the right amount of these proteins at the synapse based on how much activity there is. And then the AMPA receptors you have just the right amount of strength in your neuronal connection. And so in Fragile X, we have this problem where we don't have FMRP, so we just keep signaling through here and making proteins. These proteins are dysregulated and made to excess, and the AMPA receptors get moved in, and that makes the connection too weak. And so we have kind of a mechanism that we can work with in terms of trying to figure out how to help fix things or make things better. And this is just a study from NIH that shows that this abnormal protein in the absence of FMRP is really there. This red here is excess protein in the Fragile X mouse brain, and you can see it's right there compared to the control that has a lot less of it. So we have this mechanism, and whenever we have a mechanism, we can start thinking about drugs and compounds that might influence the mechanism. And actually in Fragile X, there have been a lot of different targets to this pathway that have been thought up. And many of these have been in clinical trials and have been used in the mouse model. And so there are other things like arbaclofen and acamprosate and ganaxolone that are in trials, that have been in trials, are also targeted to this mechanism. But what we're going to talk about today is the mGluR5 blockers, because that's what the NeuroNext trial is using. There have been a number of different ones that have been used. Some of these can only be used in mice, and some can be used in people. But these drugs block down the signaling of glutamate through this mGluR5 receptor, and the idea is that they reduce the excess signaling in this pathway that's happening because FMRP is not there to block it. So kind of, it's like a car running out of control, and this drug can hopefully put the brake on that to some extent. And so, of course, we tried that in the mouse model, and lots of labs got funding to study various things. And so there's this enormous list of mouse phenotypes that I'm not going to name off here that are reversed, but it includes things like seizures and abnormal electrical activity in the neurons, and the way the neurons are connecting, and the way the brain cells look, this excess protein synthesis, and then lots of behavioral and social and cognitive phenotypes. And so one example of this is the audiogenic seizures that the mouse gets, that are kind of like the seizures that some young children with Fragile X get. And with the mGluR5 blocker, those came way down. This abnormal electrical discharge that the Fragile X mouse has, that was eliminated by the mGluR5 blocker. Those abnormal dendritic spines or brain connections that are too long normalize when you give the mouse the mGluR5 blocker. So this has been a heavily studied mechanism, and in fact, there are over 40 papers from over 20 labs that show pharmacological reversal of about 90% of reported mouse phenotypes when you use, that's mouse characteristics, when you use an mGluR5 blocker. So there's a huge body of support that in the animal model, this drug works very well. And in fact, there's a fly with Fragile X, and you can reverse 6 phenotypes, including memory phenotypes in the Fragile X fly. And there are 6 phenotypes that were reversed by crossing the Fragile X knockout mouse with a mouse that only has half the mGluR5 receptors, and that normalizes things. And now we have a rat. And in fact, characteristics that are abnormal in the Fragile X rat are also reversed. And so this is a favorite quote of mine that one of the dads from my Fragile X clinic said, you know, when we were talking about all this stuff about the mouse, he said, well, it's a great time to be a Fragile X mouse. And in fact, we can, you know, reverse a lot of what goes on in the Fragile X mouse. But that's a simpler system than humans, and we have to be able to move to humans. So we've had some trials in humans with the mGluR5 blockers, and they haven't always gone exactly how we wanted them to. So we did an early phase, or a phase 1 trial, where just a few patients get the medication, and showed that we could reverse, we could only give 1 dose of the drug in this trial, so we showed that we could normalize a thing called pre-pulse inhibition, which is kind of a measure of sensory oversensitivity. And so that looked very encouraging. And then Novartis ran a phase 2 trial where they also saw improvement in the patients who were fully methylated. And so, but then some big trials were run that really didn't show any difference. This is the aberrant behavior checklist, and lower is better. And the placebo group actually did the best in this particular study, but the placebo group, at the beginning of the study, there was a month where there was only placebo, and the placebo group did better even in that month. So it's a little hard to interpret that. In the adolescent study, the placebo group was the same as the drug-treated groups. And this was really a study where we only measured behavior. And then Roche had a similar study where the placebo group is the middle one here. And so the drug didn't really have a big effect on anxiety. And again, we really only measured behavior in those trials. So Novartis and Roche, who had been developing this kind of drug, the mGluR5 blocker drugs, AFQ-056 and R04917523, they closed their programs on the basis of this because they just thought they couldn't treat Fragile X. But during the Novartis study, UC Davis and our site ran a couple of measures of the Fragile X phenotype, like eye tracking, where we look at whether we can improve the patient's ability to look at the eyes on a computer screen, and an impulsivity test where patients were on a computer and they only can hit the button when certain things happen, and that's called the go, no-go test. And so this is a measure of impulsivity, and patients had more correct answers and omitted things less when they were on the drug. So we think the drug has an effect. We just weren't really measuring what its effect is. And so we think it needs to be tested further. And so we've been asking the question, sort of, what went wrong in these big trials, and what's the problem with our drug development? And so once we finished these big trials that had all this basic science backing, we're kind of in this kind of circle of being a little bit lost in what to do, where we have patients and we have a mouse model, we have nice pathways, and we can fix the mouse, then we do early trials and they look good, and then we do these big trials and we do not get the result we need for drug approval. So the question is, what's the problem here, and how can we fix it? And so I think there's a couple of possibilities. And we've looked at all of these, and the Fragile X field has really kind of looked heavily on what could be the problems in all these trials. And one is that the animal models are not adequate and just don't translate to people. And we hope this is not true, because there's a lot of work done in the models, and we really would like them to be able to translate to people, or otherwise we kind of don't have indicators of what drugs to use. Or the trials didn't really evaluate the right things. And so this is a big question. When we're studying learning in the Fragile X mouse and we're not really studying learning in people, are we actually going to find the right result? So two possibilities: the outcome measures were not adequate to assess the disease, and the design was wrong and did not address plasticity or age windows of when people are learning most rapidly. And so in the mouse, we treat the mouse for a long time. In humans, we treated them for a little bit of time after they were already more than 12. And so the question is, did we really miss the window where this treatment could actually work? And is adult and adolescent behavior change really a model for changing the way neurons are connecting with each other? And probably that's not true. This is a problem from building the bridge while crossing it. No one had ever done anything like this before, and so we were going based on FDA precedents, and it turns out that may not be the best way to do things. So we need to try to fix this and figure out how to study the main phenotype and how to study the main problem in Fragile X, which is learning difficulty. We need to fix a bunch of things. We're looking at ways to have better measures of outcome of patients in a trial, like how are they improving, than just behavioral questionnaires. We need to try to go to younger ages where people are learning faster, and they might actually learn fast enough that we could see a difference between a treatment and no treatment. The length of the trial needs to be long enough to see change in disease symptoms or learning. And we may need a learning intervention to try to accelerate and focus learning. Patients with Fragile X are getting lots of therapies, but if we want to study learning, we may have to focus on one area and just try to look at learning in that area. And then we need biomarkers to show us when a patient is responding to a medication. And so what the goal is to try to see if we can design a trial that will make it a great time to be a Fragile X kid, and get away from the mouse. Well, we want the mouse to be better too. It's a guide, but we want to be able to actually move these things to humans. So the trial that I'm talking about today is a trial of one of the lead mGlu5 blockers called AFQ-056 that was made by Novartis and was in 2 of those trials in the past. And we're asking, can we use that drug and change learning? Well, if we want to change learning, we need to try to address some of the problems with the prior trials, which is that we need to go with an age group where we may have the best chance of being able to see a change in learning. And so kids who are very young are learning more rapidly than people who are older, and that's true across the board, whether you have Fragile X or not. So we want to incorporate a young age of children, do a long trial to study learning, use objective measures rather than behavioral forms where we can actually measure whether the patient is doing better in terms of their language and cognition as the main outcomes. And then to accelerate learning, we want to have a language learning intervention that tries to teach certain kinds of language learning so that we can then focus on whether those particular things got better. And then we also want to try to develop biomarkers, brain biomarkers and blood biomarkers to see if we're, really engaging the target. And this kind of a trial would then create a paradigm or a new model for the development of basic science mechanism targeted drug therapies in neurodevelopmental disorders, so that we can actually look at the effects of the drug on plasticity or learning. So this is the first ever multi-site trial studying the effects of a targeted drug treatment on learning in any developmental disorder. So this is a big thing. It's really new and novel, and we hope it's a direction that trials are going to go in the future. So we call it FX Learn because that's really what it is. And this is just Len Abaduto designed the language learning intervention, and this is patients before and after the language learning intervention and showing an increase in vocabulary after doing the intervention. So we incorporated that language intervention because there was evidence that it works. So how does the trial run? Well, people come in and they do a screening to make sure that they qualify for the trial. And then they go into a blinded treatment period. Patients are randomized 1-to-1, so you have a 50% chance of being on drug or being on placebo. And then we have a period where we just follow the patients to make sure everything is safe. And then we have a period where we titrate the dose to make sure we have patients on the best dose. In other words, the highest dose they will tolerate without side effects. And that's different for different patients. So we have a whole process to kind of get to the best dose. And then after that's all been done, we have 6 months where the patient has the intensive language learning intervention, and this is delivered with the family by Skype, because if you go to speech therapy, you only are in speech therapy like an hour a week, but the speech therapy is going to be as good as the family is at kind of carrying out all the therapy. And so really we're trying to be able to deliver a fair amount of therapy by working through Skype and Bluetooth with the family member, and then the family member delivers the therapy to the child all the time, as much as they can, in the home. And then at the end of 6 months of receiving the language intervention, we can compare the group that's on drug with the placebo group to see if the drug facilitated language learning that the intervention was trying to teach. And then at the end, everyone has 8 months everyone goes on the drug for 8 months at the end. So we can actually kind of look at the trajectory of whether the patients who are on placebo, eventually when we unblind the study, show an improvement once they go on the treatment. And then there'll be a short follow-up period at the end. So in total, there are 15 visits over 21 months, and all these little arrows show you when a visit is. The patients that can enroll are aged 32 months to 6 years. They have to have a Fragile X FMR1 full mutation. They have to have their development has to be less than 75% of normal, because we have to make sure that they're, have some symptoms of Fragile X and that they have some learning symptoms that we can improve upon. English has to be the primary language. The patient has to have some ability to do intentional communication, because otherwise they might not have enough ability to communicate for us to work on in the intervention. And they have to have stable medications and therapies that don't change. You can be on medications and have therapies, but they just can't change during the study. So we have some measures that we do throughout the study that are called outcome measures. And mainly, we have a new kind of outcome measure, which is actually a videotaped, this weighted child intentional communication score, or WCS actually, which is coded from a 15-minute sort of structured examiner and child play session. So the patient plays with a bunch of different things with the examiner, and the examiner tries to elicit different kinds of communication, and we videotape what the child's level of communication is in these tasks. And so this is an actual we're actually seeing objectively whether there's been improvement in the patient's ability to communicate. Then we have some other standard tests of language, standard tests of development or cognition, standard tests of adaptive behavior, like can the patient use a spoon or a fork, or various other adaptive skills. Then we're using the aberrant behavior checklist, which interrogates all those difficult behaviors, and a global impression of the clinician whether the patient is better or not. And then we have biomarkers with ERP and eye tracking, which we know only a percentage of the patients will be able to get done, because some kids just can't tolerate it, and some blood markers of this pathway that we are studying with the AFQ-096, and pharmacokinetics to tell us what the blood level is, as well as safety labs. We have right now 12 sites. We originally picked 14 sites. Kansas was going to be a site, but they dropped out. So now we have 13 sites from the original site choice when we started the study, and 12 of those are open, and we have 43 patients in the study. But this is our recruitment curve. So the red is the patients we actually have in the study, and the blue is the projected number of patients we need to have in the study. So we have a little bit of catching up to do, and we're trying to get people excited about the study. This just shows all of the different sites. There's a map here that shows where the sites are. I should have turned Emory to green because they just got opened up last week and are ready to enroll for all those people in the Atlanta area. UT Southwestern will hopefully be open sometime this month to cover Texas. So there are various number of the red shows you how many patients are enrolled at each site, and we are hoping all the sites are going to be looking for patients and ramping up enrollment. We have to be done enrolling to the study by the end of May 2019. That doesn't mean the study will be over. That just means everyone will be in it because of a drug expiration issue. And then we have the NeuroNext grant was re-reviewed by NIH, and so Yale is a new site that came into the network, and so they'll be they'll be doing startup activities and should be a new site within 2 to 3 months. Essentially, I am happy to answer any questions that anyone might have. I think a lot of people have called us and have had questions, so we thought this would be a really good opportunity to clarify questions about people who are for people who are thinking of enrolling, or for support groups that might want to talk about this with their families or anyone else. So with that, I'm just gonna introduce our study team, and then I'll stop talking. We have at NIH is a partner in this with us. We have Laura Mamunis and Janice Cordell from NIH who are working with us. We get the drug through the Novartis Independent Investigator Program, which I had to write a grant to. The NeuroNext team and network have been helpful in setting up. And then our key team leaders are Rondi, who everyone knows, and David Hessel, who does our eye tracking and psychology stuff, and Craig Erickson, who's been involved in our trial design and is one of our lead enrolling sites, Walter Kaufman, who's our medical monitor, Lauren Etheridge, who does our ERP analysis, Flora Tassone, who does our biomarker analysis, and this is Katie Friedman, who is our national coordinator, who is at my site and can field calls from families. And then Len Abadudo and Andy McDuffie, who designed the language intervention and have been really tremendously important in getting that rolling. So this is a lot of people that are really trying to work together to find a new way to do trials in the Fragile X field. And hopefully we'll be successful in this, but it's something we need to definitely explore. Okay, I'm done talking, and I'm gonna let anyone ask questions.
So if you have a question, type it in your column on the right, and because I don't see any questions at this point. But what I did come up with a couple that maybe that'll prompt some questions in others. Are there any medications that you cannot be on for the trial?
There are a few things that you can't be on, mainly because they interact specifically with the pathway that this works on. Most people aren't on those medications. Lithium at doses that produce a significant blood level is not allowed. And then there are a couple of other things that are glutamate medications that are often used that are used for like Alzheimer's disease and things like that aren't allowed. But in general, most of the medications that kids with Fragile X would be on, like stimulants for attention and SSRIs for anxiety and even Abilify, all of those things are allowed.
So even something like, I do know some young children who are on minocycline. Would that be preclude?
Minocycline, I don't think minocycline is an exclusion criteria.
And so is there just one person who does all the language intervention?
So the language intervention is done by a speech therapist at the site who works with the site. So there are different language people at each site, and they meet with the family by Skype. Now, we also recently put in a central SLP process where Andy can be the SLP for a family if a site has so many patients enrolled that their language therapist can't keep up with it. But in general, there'll be an SLP who works with each site and who will be doing Skype calls with the family and doing, coaching and homework sessions with the family starting from about 6 months into the trial when we really start that intensive language intervention.
Okay. A question just came in. I understand that the cutoff age for the study is 6. Is there a birth date cutoff? For example, I have a child who is currently 6, but will be 7 in a few months.
Okay, so the cutoff is while you're 6, you can enroll. So your 7th birthday is actually the cutoff. So anyone who is 6 and something, we would be happy to get you into the study before your 7th birthday.
And so it's okay if you turn 7 while you're in the study.
Turn 7 in the study. It's you have to do your screening visit before your 7th birthday.
Okay. I hope that answers the question there. Are there, I mean, I know you have lots of sites across the country, but are there funds? Like, for example, I see that, you know, in the West, there aren't as many sites. Would you have funds, for example, to bring someone in from Iowa or Utah to one of the sites?
Yeah, the West is always a problem, and it's big too. But yes, we do. We have travel money. We anticipated when we wrote the grant that about half the patients would have to travel from some distance. So we can fly people to a site. I really only talked to most of the patients we have enrolled in the Chicago area are from right near us or from Wisconsin. People have reported that their reimbursements have been fine and not really an issue. I don't know how that's going at every site, but yes, we have money to fly people into sites, and we really do want to cover the entire country. And in fact, we have said that if there's a site that kind of has too many patients and can't enroll you, and your nearest, like if you were in South Dakota and you wanted to enroll, your closest site would be Colorado, but if Colorado was not able to take more patients right at that time, we would fly you to St. Louis or Chicago or someplace where you can get in.
Okay.
So we do expect there's going to be travel.
So when does the process begin? Is it when you enroll or in May of 2019?
So we have to be done enrolling by May of 2019. So what you would do is, if you were interested, you could contact either me or Katie here at Rush, where we're, kind of the central, site that's we're managing the study. Or you could contact the coordinator or the PI at the individual site that's close to you. And then they would schedule you for a visit to come in and screen. And then once you've screened, you're in the study. What we have to do is have we need all 100 patients that are supposed to be in the study screened by the end of May of 2019. So that's not when anything starts. You start basically whenever you come in.
Okay. What kinds of side effects have been reported in the past trial, and what can we expect?
Yeah, that's a good question. That's always the first and foremost question for families, and we think about it a lot as investigators too, because we don't want to expose our patients to anything that's going to be problematic. So the nice thing about this drug is that we had a lot of experience with it in the trial, in the adult and adolescent behavior trial. And so there's been over 300 patients with Fragile X on the drug, and some of them were on the drug for up to almost like 3 years because there was an extension after the end of the trial. And so we do know that there is a kind of a dose-dependent effect that some people get, kind of like what you get on Zoloft, where if you get to too high a dose, you get a little bit disinhibited and hyperactive, and some people have trouble sleeping. So in general, that responds to a dose reduction, which is why this study is designed with the ability to adjust the dose at the beginning to make sure we don't wind up getting anybody on a dose that causes that problem. So far, I haven't seen anyone who just didn't tolerate any dose of the medication. And we have a broad range of dosing that's allowed within the study, anywhere from 12.5 to 100 milligrams twice a day. So there's actually like a factor of 10 range in the dose that we allow, and that's because we know that a lot of psychiatric medications that we use, for instance, when we use medicines like Prozac or Zoloft, we often have a broad range. Some people are on, 10 milligrams and some people are on 40 milligrams. And so it's normal for a drug that works on the brain to have a highly variable dose that's a good dose in different patients. And so we built that into the study to make sure that we didn't get people on a dose that would cause a lot of side effects. But we haven't, nothing serious has been seen with this drug. And I have to say, even in the big trial, the drug was very safe, and the main side effects are just a little bit of overactivation where the patient just starts to get kind of too hyper, and sometimes there can be some aggression associated with that if they're really kind of over the top. But we're hoping not to have it get to that point. Once you start to see side effects, then you say, oh, okay, this isn't a good dose. Let's go down to the previous dose.
Got it.
So I would say increased activity level, hyperactivity, and sleep are the main problems. And often, even if a patient's having sleep problems, taking the drug, for instance, they take it at dinner time, you can often move it to earlier in the day, just like you would for something like Ritalin, and then you don't often have a sleep issue because you were taking it earlier in the day before you were going to go to sleep.
That's pretty amazing that you've seen so few side effects, really. So here's another one. Our son is 5, and we just started a new medication. She doesn't say what it is. We're still trying to see if it works. What happens if other medications need to be changed during this long process?
Well, we're trying to avoid other medications being changed because then it's hard for us to tell what the effect is of this medication. So what we're trying to, if a family wants to, work on starting a medication, I've usually told them, let's get that, stable and then do the study. Because what we can't really, we don't want to change any medications, particularly not during the first year of the study when we're still doing the placebo-controlled part. There's some leeway for potentially changing medications once you're in the open label extension. We don't want to, but it's less of a problem for the study if people have to change medications when they're in an open label extension. So I think what you want to do is get things set. And many people, some people change medications all the time. This is true, but many patients that I have will be on a set regimen for a couple years at a time anyway, and then we'll kind of reevaluate if something new happens. But it is a problem if a new behavior emerges during the study, then we don't want to treat it with medication. So, we kind of have to try to figure out what to do to manage it. But this is kind of a feature of all studies that you're not really allowed to change your other medications that work on the brain during the study.
Right. Okay. So here, this is kind of another travel question. It's similar but different. So they're in Minnesota, but they have family in Cincinnati. Could we go all the way to Cincinnati just because we have family there?
Yeah, we're trying to be very family friendly. I think if a family says, this site's a little further away, but we have friends that we'd like to visit every couple months, that's fine. I think those choices are, we want this to be a good experience. And the difference in the travel budget is probably not going to be that huge. So I think if a family has a strong preference like that, we really try to honor it.
Could a child from Europe participate in the trial, or I'll say anyone from outside the United States?
Yeah, so the travel budget does have a limit to it. And so flying in from Europe with the number of times that we have to fly in would be really challenging. I think it would also be challenging in terms of the stress on a family. So when I have had people contact me from other countries that are quite distant, and I've basically told them you would have to move to the U.S. and live here for a while if you wanted to be in it from Europe. I don't think we could. We wouldn't be able to support all of the flights back and forth from Europe. Canada is another situation. It's usually not too difficult for patients from Canada to get to one of the northern sites. So we typically do, in fact, in all of our trials, we usually enroll patients from Canada. So that's fine. even, even Mexico maybe. But I think the flight, the international flight from Europe for all the visits would be kind of beyond our capacity. So I wouldn't say absolutely no, because I have had other families. I've had at least 3 families who've moved to the U.S. to be in a trial. I think some families have decided that they're just going to pay for flights. I mean, typically we try not to do that if travel is covered for everyone else. But again, I think that Europe is a challenging situation for us financially.
Yeah.
With budget.
Can the screening be held remotely for those who are out of area?
We can't really we have to get a blood test for the screening. So no, we can't do the screening remotely. We've tried to make everything possible that we could do remote in this trial. If you've been in other trials, you know that many trials have study visits, every week or every 2 weeks. And most of ours are a 2-month interval, with a few times when you go to a 1-month interval. We've managed to make all the drug is a liquid that has to be diluted. We do instead of coming to the site to do the drug dilution with somebody watching you, we've made that into a Skype call or or like a FaceTime call where you can do it with the coordinator and you don't have to come in. We've made all the language intervention pieces are all done remotely, but there's certain things we can't do remotely. The doctor has to see you and do a physical exam on you, and we have to get blood tests and stuff. So, and we also have to find out, what your cognitive level is, so that involves a test where the child works with a psychologist. So no, that can't be done remotely.
Okay. Could you go to your primary care doctor, or for example, there's a clinic in Minnesota, a Fragile X clinic. Could they go to that, even though they're not part of one of the NeuroNext locations? Could they go there for the screening?
Yeah, no, everything has to be done at one of the sites that does the actual trial.
Okay.
This is pretty standard with clinical trials. You can't get things done at sites that are not trained to do everything according to the procedures of the trial.
I got you. What if you have a son or daughter who has mosaicism, who is full and partial methylated?
Yes, mosaicism is allowed. Girls are allowed in the trial, and by definition, girls are all mosaic. They're mosaic between their Fragile X cells and their cells that are expressing the normal chromosome. And so what tells us whether the patient has enough symptoms of Fragile X is that criteria where you're functioning at 75% of your age level. So if you're functioning below 75% of your age level, we assume that there are enough brain cells that are impacted by Fragile X that they're causing a significant delay. And so the methylation result is not relevant in terms of that. We also know that the blood result for methylation is not always the same as what's going on in the brain. So we're not using that as a criteria at all. Anyone who has a full mutation, whether it's partially methylated or fully methylated, or whether some patients have mosaicism with some cells with a pre-mutation and some cells with a full mutation, all those patients are eligible to be screened.
Okay. Okay, here's a hard question. What happens if we see great results and then it's over? Can you give us some expectations on what could happen then? Will we see regression? I know it will take a long time to come to market. What hope can you give us, if any?
Yeah, so this is a problem with of course, any study that is not, that doesn't have an ongoing extension where people can be in it forever. So obviously patients will have to come off the medication. What we kind of hope is that, all of the work that we've done on language and with the drug being there for a while may produce some carryover and will help it so that patients just don't kind of drop off a cliff at the end. We do know, I've seen patients in the adult study who, we didn't see any kind of catastrophic stuff when we had to stop the drug. We had some patients who had been on it for anywhere 1 to 3 years in the extension. And I think we saw things that they were doing when the time they were on the drug that kind of got worse over time, but not like a sudden catastrophic loss of things. And obviously, if the trial were positive, it will take a while to get back to to do the studies that would be adequate to get the drug FDA approved. But hopefully, in the meantime, there would be some additional opportunities while we're still working on it. I don't know that I can give anything more than that. I mean, I think the key is that we can't get to the point where we have anything new without going through this trial process, and we're always going to have a situation where people are in a trial for a while and then they come out and they may have seen something really good and then you have to wait. And I know that's hard on everybody, but without going through that, we just won't have anything new. So it's unfortunately one of the necessary evils of the system.
So, but the goal would be to eventually, I mean, if the trial is successful, would be to bring this drug to market. Is that?
It would be to bring the drug to market. We don't know exactly how that would progress. We don't know what Novartis's level of interest would be. I do know there are a number of biotechs that are interested in this drug, that will be very interested in this drug if the trial is successful. So I think if this trial is a really novel trial, if the trial is successful, there's going to be no lack of interest in figuring out how to move forward. And there may be different competing entities that want to help move it forward. So I think there's not going to be an issue. There will be a time gap, but I think it will go forward if we're successful in this trial.
Another question is the trial measuring learning through improvements only in language and eye contact, or will they be looking at other data?
Yeah, so we've learned never to make ourselves too narrow in Fragile X trials. So we will be looking at the primary thing we're looking at is language and communication. We always have to say which is the main outcome that we're focusing on, just because that's part of trial design. So the main thing we're looking at is improvement in the communication in areas that we're going to work on in the language learning intervention. But we do have a bunch of other things on that list that I had here. So we're looking at global cognition. The Mullen is a test of all different kinds of thinking and learning. We do have a number of different language tests because, again, that's what the trial is focused on. Adaptive behavior is very important. The Vineland is just a measure of how the patient gets by in daily life, and it's turned out to be it's a good measure of just functioning. And so we are very interested in those measures. Those are all secondary measures, which means that that they may not be the primary, but we know that they're very important indicators of function in Fragile X, so we will be looking at them closely. And the ERP biomarker is a direct marker of brain activity, so it shows us if the EEG activity in the brain has changed with the drug, and so that we'll be paying attention to, as well as the eye tracking gaze. So we're casting a broad net, even though we're focused on language.
So going back to the when the trial is over, could there possibly be an extension of this trial? So let's say your things are going well. So like right now you have it where everyone goes on it for 6 months.
8 months.
8 months. I mean, could that part of it ever be extended?
Well, not right now with the way Novartis made the drug, especially for this trial. So they don't have batches of it just sitting around. So that part probably can't be extended. There'll probably be a situation where we have to finish the trial, and different people are going to finish at different times. And then we have to gather all the data. So we have to at least finish the placebo-controlled period and analyze the data. And then if we had a successful result there, I think we would really start brainstorming with Novartis about what the next steps would be to do a new trial or to provide people with access or what. But I certainly cannot say that we know exactly when we would be able to offer someone more treatment after the trial.
Okay. Here's another one. My son is 6 and is totally nonverbal. He is using an augmentative communication device. Would he still qualify under the communication?
If he has acts of intentional communication, which it sounds like he does, if he's using an augmentative communication device, he would qualify. And in fact, we even have a special process for people that communicate mainly with their augmentative communication device, where when they do this, if the person uses their augmentative communication device, if that's their predominant way of communicating with their family, and their SLP has decided this is the best way for them to communicate, then they can use the augmentative communication device, and they can embed it in the intervention and use it when we do this play and communication assessment. So yes, someone who is nonverbal could qualify for the trial, and depending on exactly how much and what ways they're using their alternative communication device, they may use that as a part of the study to measure improvement in communication.
Okay. Let's see. My list of questions is kind of funky here. Just a second here. What is the first step to enroll in the study?
You can email me or Katie, or depending on where you're located, you can email the study site. The study sites are all listed on clinicaltrials.gov. And then the NeuroNext website, I believe, has them as well. But if you really want to just find out who to contact, I think Jane is going to give you the email for either me or Katie, and we can direct you right to where you would need to go. And then patients that contact us, we're going to kind of keep track and make sure that you get into a site that's the site that you're interested in relatively quickly.
Okay, let me see if I can post this for everyone. Send to all. Okay, so everyone should be able to see that now, I think. So you can see Katie's email and her phone number. And also, when this is over, Dan will send a follow-up email to everyone with the contact info, our question and answer page and recording of the webinar, etc. So that will go out too to everyone. Let's see if I have anything else here. Okay, someone else is asking about the contact information. So you can contact Dr. Bray Kravis or Katie or call her. And let me see, I'll post that again and I'll see if that comes up and I'll send that to all. So that, I think that's showing up, but we will follow up with that.
Okay. Yeah, and okay. I think you, somewhere on the national website, you have it posted too, right?
Yes, we do. We do. Here's another question, and I know there was a slide on it, but it went through. How many times do you need to physically go to the site?
Okay, so every time there's an arrow here, you have to go to the site. So it's 15 visits over 21 months, which is less than a visit a month. So that's pretty. It's a long study, but it's less frequent visits than most clinical trials would be. So you do actually have to go a fair number of times. And I know that travel is a certain level of burden, and it depends on the family and how well travel goes with your child. But this is what's required. In between those visits is when all the phone stuff goes on and Skype stuff.
Okay. This is similar to one of the previous questions. What if your child is semi-verbal? If you want to reiterate that criteria for
semi-verbal is fine. The weighted communication scale, that's our main measure, measures communication. It measures nonverbal communication as well as verbal communication. So it's not a problem for patients who are semi-verbal. Certainly, anyone who has some words is very likely to qualify. The only situation where patients may not qualify is if they're really not using any verbal or nonverbal communication to intentionally try to communicate with the family, and that's most Fragile X kids are not in that situation. So you don't have to be verbal to be in the study, but you do have to have some ability to indicate when you want to communicate.
Okay. Okay. So we're getting close to the top of the hour. Is there, and I think we've answered all the questions that have come through, at least for right now. I keep double checking because they're not showing up in order. They're just showing up randomly. Here we go. Oh, so this is not a question, just praise. Thank you all so much for doing this. We need you. So that's very nice of you to say that. So are there any parting words you'd like to say to, additionally encourage families to consider this or at least reach out for more information?
I think, I think I've made the points that the main exciting thing about this is that we're trying to accelerate learning, and really none of the other trials in the past have done that. So this is a new idea, and it's a step on the way to trying to develop drugs that will improve learning as well as behavior. So we're very enthusiastic. I want to thank whoever just sent that last message in, because this is a critical, it's a critical kind of trial and thing we need to do in the field to move to kind of the next step of treatments. And they're going to be, things that have to be smoothed out, because whenever you do something the first time, it's always a bit of a challenge. But I think it's really important, if you can do the trial and you're interested, we will bend over backwards to do anything needed to kind of help and support you. So we'd like to have lots of participants, and I want to reiterate that this is a trial about learning language. So you don't have to already have language. The idea is that hopefully by being in the trial, you're going to gain some more language. So, and if not from the drug, it's from the language intervention. So we are very enthusiastic and are happy to help anyone who wants to move forward. And I know that half the planet emails me anyway, so if you wind up emailing me about it, I'll just wind up sending you to the site that's closest to you, or we'll wind up getting you in here. So I think that's really all I have to say.
And
I want to NIH and the NeuroNext network also, because they've been tremendously supportive, and this is not a small enterprise, this trial. So they've been tremendously supportive in getting this going and keeping it going.
If you could maybe just one more time, if you could put up the inclusion criteria, because one woman is asking, what if your child's CGG repeat is 54?
Oh, okay. Well, 54 is a pre-mutation. So if you can have a 54 if you also have a full mutation, but the pre-mutation is that we don't consider that to be Fragile X syndrome. So that somebody would not be eligible with a repeat of 54.
Okay. Well, I think on that note, we'll end this webinar. Please, we'll be following up with additional information. If you have any questions, or if you want to email me, please email me or Dr. Barry Kravis or Katie, and we'll get your questions answered. Just to echo Liz, we appreciate you all, all your interest in the trial, and we hope that we're able to make it work for your son or daughter.
Okay, and I want to thank the foundation for hosting this and for also supporting us in this trial.
It's always a pleasure. All right, thank you, everyone. Have a great rest of your day. We'll see you next time.